I'll turn it over to Rob so he can introduce our speaker, Dr. Ryan Harrigan.
Well, I think everyone has probably already met Ryan by now, but Ryan Harrigan is at the UCLA. He's a adjunct professor and researcher, and I've known Ryan for probably over 2 decades now, but we've never actually worked together until recently, so I'm quite looking forward to that. And it's really nice. I've learned a lot about what Ryan's been up to this summer. I guess he had a really good Brett summer, and I'm kind of really glad that he took time off and decided to come here today and talk a lot about his genomics work that he's doing. I think everyone will be really impressed and kind of really interested in what he's been doing and with the conservation twist on using genomic data.
So can I advance with this? Hey, everyone. My colleagues are, oh, there it is. What was that with the mouse? Okay, I got it. We're good. So organized today. I mean, no, I have a pointer. Thank you. Hey, everyone. Good to meet everyone. If I haven't met you already, thank you to University of Nebraska. It's my first time in Nebraska. It's a lot of fields. From Los Angeles. I grew up in Boston. I've always looked this way. I don't do any drugs. I didn't get this way by living in California, whatever you heard. I've always looked like a hippie. So just bear with me. Thank you to my hosts, Rob and Sarah. Sarah, thank you for teaching me how to feed a horse yesterday. She really thinks I'm from the city. Okay, I'm going to talk to you guys today about work that I've been doing over the last 8 or 10 years. This is work that I've done with colleagues, of course. These are not independent projects. Science is not a DIY. You got to rely on your collaborators. So I want to, acknowledge and thank everybody that I've worked with. I've probably forgot some people here, but it's a lot of folks that come together to produce a paper to do good science. So I'm in no way taking credit for all the work that you see here. I'm just honored and privileged to kind of be presenting this work that I've worked on with other folks. Motivation for a lot of what I do is, we live in a really great time to be doing science. We have tons and tons of genomic resources. I'm going to sound super old right now. I'm that guy that's like, I remember when I used to have to pour my own sequencing gels and run 36 lanes at a time. I did do all that stuff. You had to knock the acrylamide and everything. We don't have to do that anymore. You can get these vast amounts of whole genome data or genomic representation from species very quickly, relatively cheap. The advances in computation, I hope to kind of convince you all of this by the end of the seminar. There is just a massive amount of computational power in your laptop computer that we really didn't have access to before. That allows us to run these kind of environment genome models really quickly. On top of that, we have all of these citizens or resident science data sets. So people are out collecting more data than has ever been possible before. I did an eBird search. Everybody familiar with eBird records? Did an eBird search for LA before I left. For the 2024 year, there was 4 million records in Los Angeles County. So there's a lot of people looking for birds. That, of course, comes with perils. There was an ostrich reported. Probably not an ostrich. Somebody mistyped something. Maybe an osprey. Who knows? Who knows what was going on? They may have been drinking. But you can vet those things and filter those things and get gobs and gobs of data. And on top of that, we get environmental layers from a lot of different sources now. So remotely sensed layers from space, all of those are prevalent. And my motivation was kind of like, well, we have all these really cool data sets. How can we kind of combine those? And not like in a nerdy look at my model way, but like in a real informed, applied conservation way. So I've always been kind of routed in, I got my PhD doing duck conservation with Rob. We've always been kind of rooted in the idea that the model and the analyses we do should be, able to inform real management and conservation decisions. We have rapid anthropogenic change, however, though, and I love this infographic. I show this anytime I can. These are, sorry, turn this on. No. Did you say there was another pointer? Wait, there it goes. we live in an unprecedented time in terms of change. This is our year right here. These are global mean temperatures from 1850 to 2200. And so if you look at the past average temperatures, despite what some people report in the news, the planet is warming for sure. And we're right here. This is representative concentration pathway 8.5. I don't know if you know those concentrations. They're like future projections for scenarios. Guess what? This is the worst case scenario. This is our best case scenario. So even in our best case scenario, we are living in a time where we are about to experience, and wildlife, organisms around us, the planet is about to experience all of this accelerated change. And so as scientists, the idea is, okay, with all of these tools we have, with all of this data, how can we prevent this from happening, have this happen, but then when we go through this lower best case scenario, how can we make sure species can come along with us? And I mentioned this. What can we do with all of these data? How can we get these genomic signals into a relationship, or how can we determine the relationship with environmental conditions? And then how can we apply those to real conservation decisions, something what I've been doing. We can also do this really cool thing when we link it to environment, which I hope to cover today. When we link those genomics to environment, we can also then say, okay, what was the environment like in the past or in the future, like the slide I just showed you, and predict or project back as to what those relationships look like either in the past or what they may look like in the future. Why does this matter? Well, because under future climate change, we know species are either going to die, they're going to move, or they're going to adapt. And so understanding how the move or the adaptation might take place helps us with conservation. I'm going to cover 3 types of data today, and this is just the way I think of this. This is not the only way to group these things, but it's how I've kind of approached my science. And I'll cover these bottom 3 today. I won't cover non-spatial data, but listen, non-spatial data is super cool. We have a solar project right now in Southern California looking at how bird populations are affected by the solar industry. 30% of the birds that are recovered from solar panels are kind of fried and singed or can't be identified because it's only a remnant. We take those data, those samples, we run DNA tests on them. We don't really care about the location of those samples. We just want to ID the birds. That's an example of non-spatial data. Don't get me wrong, all of that is amazing. But if you can get spatial data, why not? If you can include spatial data in your analyses as you're developing, let's say, your graduate school work or even your undergraduate work, if you can include spatial data and spatial data is available, I hope to convince you that that's a thing to do, that you should do. And here's the 3 things that we'll cover. So we'll just talk first about using spatial data to understand species, not really modeling anything, just including spatial data when we look at populations. Then we'll go through a couple examples modeling us. I said a single thing, pretty general, but it was on purpose. I want to just cover in a general sense, if you had any kind of character, genetic, morphological, what have you, song, which is a type of morphology. But if you had anything, how can you map it or model it to environmental conditions? And then we'll get super nerdy and model several things with environment. And that's where the science really gets exciting. All right, here's using spatial data to understand species. I mentioned this can be based on any kind of interspecific character. We can use the location of individuals and their genetics or their morphology to kind of justify conservation units or even explore hypotheses. So this is often used in conservation. How is conservation done? By mapping out a certain piece of land. Well, if that certain piece of land doesn't match the biology of the organism, then you're really not conserving that population or that species. And so the idea here is to use that spatially. By the way, if you're trying to sell conservation of something and you have a map of it, it is just wildly easy to do. If you say, here's where the genetics of this organism lie, and you show that to a planner, a management planner, or somebody making a decision maker, it just works. If you're showing them DNA sequences, you have failed. It's just not going to go over well. So the idea of being able to use these things and present them in a form that everybody understands, a map, it's a powerful thing. And as I mentioned, we're not really modeling any relationship here. We're not saying here's how the genes or the morphology is related to the thing's environment. We're just looking how they're spatially distributed on a map. Hopefully you'll be able to see that distinction. This is not terribly sophisticated, but the writers, the authors of Guillot et al., I mean, it's amazing that they were able to kind of just to convey this accurately. Previously we thought of, these are two species, by the way, a red triangle and a black circle. In our brains, I think we always previously kind of thought of like, oh, maybe this is how populations look on a map. Maybe they're just sort of randomly distributed in space. And you're collecting genetic data. If you didn't have any spatial data, you might think, oh, this is pretty reasonable. But we all know, everyone in this room, that that's not how populations are actually distributed on a surface or across a region or an environment. In fact, it looks much more like this, where you have red triangle individuals that all have something in common, gene, series of genes, morphology, all kind of seem to be in one portion of the area, and the black circle species seem to be in another. And we don't know what those patterns will be. We also don't know if there won't be breaks in between those, but we do kind of think that individuals that have a common morphology or a common character or trait, common genetics, tend to map themselves out in the environment like this. Here's a fun example, near and dear to John Benson's old stomping grounds. This is a side blotch lizard. This is, oh, I should say, oh no, I put a map up here. Here's Los Angeles County. I live right there. The Kardashians live somewhere in here, just to kind of place this. So this is a very rich neighborhood in Southern California. You can see the scale of this, very small. But this is the 101 highway, which is a 5-lane highway in both directions. I mean, it is massive. It's a pretty major highway throughout most of California. Here's the PCH, which is the Pacific Coast Highway. That's one of the most beautiful. If you're ever in California, drive on the PCH. It's unbelievable. You have the beach and the ocean right next to you, Pacific Ocean, and then to your left, or sorry, to your right, you have just a beautiful mountainscape. Anyway, we collected side blotch lizards. This is a project that we did with National Park Services from all over this area, and we just said, hey, let's look at the genetics of these things and determine how those things, again, cluster in this environment. Not looking at anything else in the environment. Let's just see how the genetics play out if we map these. And here's what we found. These are two populations. They break down into two populations where, check this out, the purple population only exists north of the 101, and the greenish population here only exists south of the 101. Now, we haven't mapped this to environment at all, but look at this. This green population here is more similar to this green population over here in terms of its genetics than it is to this population here, which is purple. Now, you don't have to be a rocket scientist to say, well, what's causing that? As I mentioned, you have the 101 running right through that environment. So this is kind of a real world example of how an anthropogenic event, the building of that highway, has sort of separated these two populations. If you're still really paying attention, you'll see there's this one population here that's south of the highway. And we were so confused. We're like, flying lizards? What is it? It's the flying squirrels of Nebraska, right? Like, how did they get across a 10-lane highway? And we were so confused about this. We met with National Park Services, and they said, there's an underpass right there where that 101 sign is. And they've also had camera traps set up where they saw those lizards and other organisms using that underpass. So this was like, we had really no idea what was happening. And then it's like, we meet with the Park Service and they're like, yeah, there's an underpass there. So that makes perfect sense. So we do have some crossing of that major anthropogenic event, but not much. And that's what separates those populations. This is a wren tit, which is a bird. Nobody thinks of birds being sort of not able to cross the highway, but wren tits are kind of like the chickens of the small small passerine bird world. They're really terrible flyers. And I don't know if they'd be super successful at getting across the highway. To sort of prove that point, we looked at their genetics, and you have this north population and the south population sort of completely separated by the 101. So we're seeing that this anthropogenic event, the building of this highway, has very much influenced the distribution of genetics and likely morphology in this area. We looked at five other species, including bobcats, foxes, and a couple other herps, and see very similar patterns. So those are all in the paper. I just wanted to point this out because it's a cool kind of local scale story. And it's relevant because we have this brand new Annberg overpass that's being built. This is the largest wildlife corridor that's ever been built on the planet. This was recently approved. They've begun construction. It's like 82 girders or something like that. Most of those girders are up now. I know it doesn't look super impressive here, but I did the math. This is about a half of a US football field. So about 50 yards in width and across this major highway. So what does that do to the populations? Well, we don't know. We're hoping that things like red tits and side-blotched lizards will be able to move across that, and we'll get some more, genetic mixing and integration between those two populations. There's a regional scale example here. This is a paper that I co-authored with John on the eastern wolf. Of conservation concern, the eastern wolf is larger than a coyote, but smaller than what you consider like the big gray wolf that you see out in the West or in Alaska. And nobody really wanted to, people are like, oh, we don't know if this should be a distinct thing. It interbreeds with both coyotes and gray wolves. So can we really justify the conservation status? Also, what if there's a hybrid? Do we conserve that, or do we treat that the same way? And if you look at the genetics, well, the blue is the eastern wolf. So most people in the room have seen these kinds of PCA plots with admixture results. And it turns out that the eastern wolf is its own thing, no doubt. The coyotes are in red here, so there is some interbreeding. You can see blue in here, and the gray wolf has some blue. But for the most part, that's as distinct as any one of those other categories. And if you map that out spatially, you can actually find the area where you see eastern wolves. Super valuable and critical for building a conservation message and saying these are the areas we need to conserve to make sure that this eastern wolf, this unique Algonquin wolf, they call it in Ontario, is preserved, and we can conserve that population. I heard yesterday that John's working on like a new model of looking at how those integress and stuff. So this is the impetus to finish that model up because I want to hear how this story ends. Okay. I'm going to go to a continental scale here. I would be remiss to not mention the bird genoscape project. This is my colleague, Kristen Rug, her lab at Colorado State University in Fort Collins. She's doing a lot of this work on these bird genoscape projects, and I am basically a helper on this, but we've been thinking about these problems for quite a while. This is a flycatcher paper that we wrote, and this is on the continental scale. So if you look up the range of a willow flycatcher, it shows all of this is the breeding range. They don't do these nice little breakouts, but when we look at the genomic signal of this bird, you actually see very clear population structure, what we call nodes. And these nodes are really important to identify. Why? Well, we could manage or conserve things in different ways. So for the willow flycatcher, there's plenty of concern about the southwestern willow flycatcher, and we might want to conserve or manage those things differently than one of the other subspecies. Another thing to keep in mind is when you do these kinds of analyses, you can see that obviously there's some large nodes and some medium nodes and some small nodes. In a lot of these cases, the smallest geographic nodes harbor just as much genomic variation as one of those larger nodes. So like bang for the buck, these small conservation regions are super, super critical, because if you lose one of those populations, you're losing a fifth or a sixth of your total genomic variation. Why do we care about genomic variation? Because that's the only thing that we have to combat climate change. And moving forward, these things need to have interspecific variation to deal with rapid climate change. Just going to show you this really quick. This is a study we're doing on the solar project. So looking at the effects that solar energy industry has on bird populations. I just want to show you this because this is a practical example of how we use these genoscapes. This is 5 species. I know they're small. The kestrel has always been a problem, moves a lot of different places. Like these genomic groups were not easy to identify. We actually had to dig pretty deep into the SNP data set. But I mean, check this out. This is a common loon. There's a red area here and a yellow area here we call Midwest and North Central. If we look in California at loons that struck solar panels in California, you can see red and yellow only down here. That means that loons were found in Southern California that are eventually going up to these Midwest areas to breed. They're following the coastline up, and if you don't know California at all, there's a giant reservoir called the Salton Sea there. It's a stopover site for a lot of migratory birds. So these loons, we think, are coming up from breeding grounds and actually taking a break in around these areas susceptible to solar impacts, and then making their way over to the Midwest. So these genomic or genetic nodes and groups get more complicated when we think about migratory birds. Now, why do I care about loons in Southern California? Because if that's a bottleneck for the population, or those populations are being affected, then they don't ever have a chance to make it back to breeding grounds. Migratory species are notoriously difficult to track, to understand their full life cycle, and to kind of understand how they change with the environment. But in fact, migratory species do a really good job of just tracking environmental resources. So, I said this looks like I do LSD. I'm sorry. This is going way too fast. We did a conversion really quick. But this is showing you the size of these continents are proportional to the productivity during different months. So I love this animation because this is really, we always see just a static map of the world because our human brains do well with that. But this is the actual resources available in each one of these compartments throughout the months. And so you can see why maybe neotropical migratory birds might be following resources in South America during wintering times and, you know, go up to breeding regions for new resources that open up during breeding times. And so thinking about how these animals are tracking these resources is something that we've kind of been obsessed with. Eric Anderson is at CSU as well. He works with NOAA and other groups. He does these really cool animations. I'm just going to play this. Let's see how it goes. Yeah, this is going. Watch these months. Once we develop these genetic nodes, we can see where because we can ID them genetically, we can see how these nodes fill up over time. And so you can see during the breeding season, A, how big those nodes are, but B, where those nodes fill in, what time those birds arrive there. And then as the fall hits and birds start wintering, there's be a little bit of a break here and you'll start to see them popping up on the wintering grounds. And again, this is for that willow flycatcher that I was mentioning before. Okay, yeah, we have missing data, sure, but we're trying to fill it out. You collect birds down here, you genetically ID them, and you can track them to those breeding nodes that we previously identified. So we're getting there. We're getting close to being able to identify full life cycle biology of these organisms. Why do we care about this? Well, breeding nodes and saving lands in North America is fantastic. But if a bird only winters in a small portion of their Central America, and that's lost to crop conversion or anthropogenic activities, there's very little chance that that node's going to persist. Here's a less video-ish version of that, where this is the Wilson's warbler. We have these breeding ranges. I just want to point out, this is so small. There's a coastal California population here, again, that harbors as much genomic variation in this population as all of this Western boreal population. So we care about the small stuff for sure. And you can draw what's called migratory network models, which shows not only the connections between breeding grounds and wintering grounds, but it also shows the strength of those connections by the width of those lines. Let's talk about this. We'll nerd out about it later, but I'm going to run out of time if I keep going. Southern Baja and coastal California. The only place we've ever seen coastal California Wilson's warblers breeding is, the only place we've ever seen them wintering is in Southern Baja. So, and one little sliver here in Western Mexico. So to my point earlier, if we lose that little sliver in Southern Baja, those coastal California birds have no place to winter. And do they move? Do they find another place? Unlikely. I mean, these are evolutionary trajectories that they've been taking for lots and lots of generations. I want to brag a little bit about a postdoc student, or a former grad student, now postdoc, Matt Desai, who just wrote this package called Mignette. Do you get it? It's hilarious. John, it's like a vignette, but he was holding his head like, oh, yeah. It's a vignette, but it's migratory networks, so it's now a mignette. Okay, we were really proud of that. The reviewers were not as proud of that, but we got it through. It's called an R package for creating visually migratory network models. I put 2024 question mark, that's not a typo. It literally just got accepted a day before I got out here. So I don't know how the publishing thing works. Maybe they'll have it in by 2024. If not, early 2025. This is so cool. So he's got a whole, Matt developed a whole R package that you can go through a whole tutorial for any migratory species. It doesn't have to be birds. And what's more is you can use geolocators, or if you have isotopic data or any other kind of tracking data, you can use that to track animals and create these wonderful looking networks. He even got super fancy and did these alluvial plots. Ooh, I love these things. So these are showing the same kind of migratory connectivities, but with the width of those things being the width of the strength of the connection. And this is for American redstarts, osprey, and Swainson's thrush. Those examples are provided in the paper, so you can take that data and run through, if you like to nerd out and stuff, you run that through the whole R code and produce those same kind of, they're called alluvial plots. I mean, you can use those for anything you want. We don't have a copyright on them or anything. This is the cutest animal I've ever worked on. Are you kidding me? This is a pangolin baby riding on the back of its mom. That's how it moves through the forest. These are African pangolins. Why am I showing you this? Because you can apply these same types of methods. Again, where do we see pangolins? What are the groups? What are the genomic groups that we see? The reason we were able to do this is because we were able to sequence the full genome of a white-bellied pangolin. It's the first time it was ever sequenced. We also got a transcriptome and did the whole number of chromosomes. We did the full genome and figured out how many chromosomes. New claim to fame, this thing has 114 diploid chromosomes. It's the second most of any mammal and the first most outside of the rat family. There's a rat, stupid Gambian rat had 118. Otherwise, it probably would have been a science paper. Damn. Anyway, 114 diploid chromosomes. So really weird chromosome structure, particularly because 4 species of Asian pangolins have between like 32 and 48 diploid chromosomes. There's 8 species of pangolins total, 4 in Asia and 4 in Africa. The 4 in Asia have been largely decimated for Eastern traditional medicines. They don't work. I'll say it on camera, record it, I don't care. They're not doing anything. You don't need pangolin scales for anything. But they're being harvested, and once the Asian species got wiped out, they're sort of moving into Africa and illegally exporting these things. All of these species are on the endangered list. And the cool thing about this is not only are we able to track where they are in Central and West Africa, but because we have the genomic profile of those scales, of those individuals, we could get illegal confiscations in Hong Kong and genotype those and figure out where in Africa they came from. So we're seeing a much larger, if you look at just the seizure list, it looks like everything's coming out of Nigeria. But in reality, if you look at the genomics of it, these pangolins are getting moved from all over Central and West Africa. And it turns out they're getting probably loaded onto cargo ships in Nigeria. But that's not the full story. Somehow there's within country movement in Africa, moving these pangolin scales out of Africa. We'd love to do this, continue doing this, funding pending, but for the other 3 species, because oftentimes when you get seizures, you get a mixed group of pangolins. Okay, everything I just showed you was, again, looking at the genes or the morphology of a thing and just placing it on a map. without any regard to the environment it's in. But now I want to talk about actually modeling things to the environmental conditions they exist in. I'm going to talk to you about random forests. Can you just show of hands online? I mean, it doesn't work online. That's why I made that joke. Show of hands in the audience. How many people have heard of random forests before? Okay, it's like a good number of you. I'm going to do this in the most basic form. Just if you remember anything, this is the slide to remember. These random forests are models that link a thing. It can be anything. It can be a morphological character. It could be a SNP. It could be bird song. Why would I say bird song unless I had a slide coming up that showed bird song? It can be anything you want. You're trying to map it to the environment that it belongs in or occurs in. And you do that by just taking any one of the environmental predictors. I just called this predictor 1 for funsies. And it just says, hey, if you have a high song frequency and a high song frequency and a low song frequency, how does that environmental variable split that up? How can I just put these two things into a group where this whole group has low song frequency and this whole group has high song frequency? It's just looking for the split that best puts those two things into a bin. And then what it does, which is really cool, is it keeps doing that. It keeps trying to split your data. So here's two other predictors. This might be temperature. This might be precipitation. This might be land use change. It can be any predictors you want, any continuous or binary predictors. It keeps going along and along. I purposely left out some numbers here. Don't be alarmed. I also purposely put in predictor 1 again because it can use a predictor multiple times. You might have a big portion of the reason you have low song frequency and high song frequency be mean temperature. But then in a much smaller suite of the data, temperature might become important again and allow you to split that even further. So it doesn't care if you're using the predictor more than once. It doesn't care if it includes some predictors and leaves others out. You can see here, I don't have predictor 4. I have predictor 3 twice. That's fine. By the time you get done with this, you have bins of responses. This is your song frequencies. And you now have a way to describe the locations in which you see low song frequency, high song frequency, and some intermediate values. And that's one run of the forest, and they call that a tree. That is a single tree, makes sense. But the reason I repeated predictor 1 here, and predictor 1 here, and predictor 3 here, and predictor 3 here, is because these trees are run, are you ready for it? Randomly, which is why they're called random forests. So you're randomizing your predictors every time you run the tree. In addition, you randomize the data that you give each one of those trees. So you're withholding some of your data in true iterative fashion. You can leave some of the data out. And guess what? The model is only as good as the data that you can predict, the data that it never saw. So it trains on a certain suite of subset of the data, and then it tests on data. And the data that you test on, if it does well at predicting that brand new data it's never seen before, then that particular tree and those particular predictors score very high. If it falls apart when you give it new data, why would we want to use that as a model? It will tell you this is not a good predictor of this particular response. And so that's a really attractive feature, and it's really what separates this from a linear model. When you run a linear model, I see them all the time on posters, in presentations, in publications. When you run a linear model, you're using all of your data all at once, and you're fitting a line to it. And great for you. That's amazing. You won the science fair. But it's not great if you want to say, hey, in new places that the data has never been observed, is this model going to hold up? This is boring. This is so boring. Why did I put this one up there? This is just a single tree. The reason I put, there's no quiz. There will be no quiz. This is just how these trees look when you produce them. There's another nice feature which I didn't mention, which the length of these bars usually indicate how powerful that predictor is. So the larger the predictor, the more important the predictor is, the larger these vertical bars will be. Also, the first split is always the most important. So in this case, again, no quiz. Quick scat is surface moisture. This happens to be a tree where surface moisture plays the most important point in the most important predictor in describing this relationship. The reason I showed you this tree is because it's awful. I mean, it's really, you have to interpret this, and you have to do that 5,000 times if you're looking at a random forest. That's typically like how many random forests you would run. So really impossible to interpret that. Not a good way to present data. A look in supplemental information for 500 trees and my evaluation of each one of them. So we have to come up with another way to sort of look at the results of this without looking at this kind of boring tree. So we have another thing that's reported as part of the random forest. Oh, by the way, like talk about computational horsepower. Random forest, you can run 5,000 trees in like 22 seconds. It's so fast on even a crappy computer. It's unbelievable. It's just pouring through all of that stuff. If you think humans are going to be around in 50 years and we're not all going to be AI, just run a random forest and you'll be like, oh man, we're really in trouble. These things are just ultra efficient. So we get these measures of variable importance. I purposely took off any of the names here because I just want to look at overall patterns. In this particular example, which is a real world data set, you get 5 of the top, I don't know, however many, 22 or 24 that are here, are temperature. So again, you're running all of these together. You're running all the temperature variables together. But remember, in each one of your trees, some of these predictors weren't in all of your trees. Sometimes they got left out. If they got left out and your model dropped in performance, big warning sign. That means you really need that predictor in there. So you don't have to, when you're running your random forest, you don't have to worry about collinearity. You don't have to worry about things like, oh, covariables. Oh, maybe there's interactions between. It's gone. You just run everything in there. Everything but the kitchen sink, throw it in there and see what rises to the top. Another very attractive thing. When you see temperature like this all at the top, you say, okay, it's clearly temperature is an important predictor here. Now, I don't know the exact relationship between temperature and my response variable. That still requires, guess what, ecology, like good ecology and good hypotheses. So you have to have some understanding of why temperature might be important, but the model's not going to tell you that. You have to know that. There's a famous example where you can predict like the number of gulls in a city by number of McDonald's restaurants. So like the proximity of McDonald's to the individual. And so like, okay, they're not really attracted to arches. It's more that they love French. Who doesn't love French fries? So that's a food resource. Again, there's a hypothesis, there's ecology there. But if you just looked at number of McDonald's, you could predict gull presence. Okay, here's the song frequency example. This is a little small innocuous bird called the little green bull in Africa. From space, from remotely sensed layers, we did not do, initially we did no field work. We collected the samples, the song recordings, but we didn't go out and measure any environmental conditions or anything like that. From spaceborne data, remotely sensed layers, we are able to predict birdsong. I can tell you what frequency the little green bull is singing in Central Africa. As you move down into this forested area in Cameroon, you see the little green bull, same species, those populations sing a higher song frequency. Whereas the more, this is like a drier area, more savanna area, you see them singing a much lower song frequency. Any like participation time, any guesses why birds might change their song frequency to higher in forested areas? Is there any wild guesses out there? Yeah. I love this. Sound is like bouncing off the trees, the physical acoustics of the environment. Maybe females can hear the birds better. Oh, a fantastic hypothesis. Any others? Okay. We had a couple more, like maybe it's the sound, the acoustics of it. Maybe the temperature differences. There's temperature differences between them. Maybe there's something going on. We were searching and searching and searching, and then there was another study on tinkerbirds that from a completely different area that showed that tinkerbirds were singing higher frequencies only when cicadas were singing. There's 2 species of cicadas here that are deafening if you've ever heard them. And it's the exact same frequency that the lower song 3 maximum is sung at. So we're thinking evolutionary. We don't know this for sure, but we're thinking evolutionarily, there's those 2 species of cicadas are forcing those populations in the forested areas to just go up a little pitch so females can hear them, you know? It's like, check it out, I'm not a cicada. Super attractive male. We don't know if this is true, but we had this model in hand, and then we went out into the field, really cool, and we were able to ground truth on these 2 sites. We went to these 2 sites, we hadn't recorded them before, we went out with a recorder and got their exact song frequency. So despite all the nerdy random forest stuff, we were able to go out and say, wow, we nailed what that song frequency would be, even though we didn't go out and have to measure, you know, every one of those sites. We could actually do it remotely. One of my favorite examples of all time. It's a zebra, or if you're from South Africa, zebra. People pronounce those zebras in South Africa. I was confused too. The zebra has stripiness. That's a real word. We used it in a publication. They have there's a stripiness. And they have hind leg stripiness and torso stripiness. And as you go from North Africa to South Africa, in the plain zebra, again, single species, you can see, I mean, this is like top shelf where mama hides the cookies zebra. This is like as stripy AF, if you know what that stands for. Really fun zebra. Hind leg stripy, torso stripy, good times. As you go to south, you get less and less stripes. So you go from a very stripy zebra, same species, down to this thing that almost looks like a quagga in the southern part. We had no idea why zebras were getting less stripy. There were a number of hypotheses, hint, hint, as to why zebras would even have stripes. What do you think? Any weird, wild guesses? John Benson. Right, so mosquitoes and tsetse flies, there's some reports put out that flies and other aerial insects have trouble landing on black and white surfaces. Like that, if it's contrasting colors, they get confused and can't land on the zebras. Love it. That's H1. We're like in undergraduate science now. What's another one? H2. Any other ideas? Come on, don't be shy. Yeah. Okay. Another report came out that said white skin was cooler, black skin was hotter, but the cool thing was the contrast of those creates small eddies in the air temperature around those zebras, and it's like you're walking around in air conditioning. Thermoregulation. There's one more that was like pretty prominent. What do you think? What else do you think? Did you say disruptive? Like they're like fighting the man? To avoid predation. Yeah, so lion predation was a big one, right? Disruptive colors so that lions can't pick up. Lions, by the way, really good at picking off zebras, even the really stripy ones. They're really good at it. I mean, amazing at it. So we didn't know. We run a random forest on this. I'm here to say, is this being recorded? It's temperature. It's totally temperature. You can predict how stripy a zebra is by the temperature in that area and the temperature they exist. The lion thing is appealing, alluring. When the zebras were evolving their stripes, there's some evidence to suggest that there really weren't that many lions in a lot of those areas. The tsetse fly thing, I'm not making this up, you can look it up. Somebody just came out with a paper that said it's definitely tsetse flies because they put some flies in they did striped bottles and saw that the temperature differences weren't real. And they said it's got to be tsetse flies. I don't know if I believe it, honestly. I'm going to stand on the temperature thing until I'm 100%, because this random forest model, we included everything in it. Tsetse fly distributions, lion predation maps, temperature, it's just temperature. The temperature is describing why we see stripy zebras. Do we know for sure? No, but guess what we have? Remember old people in the room, Flickr? Remember that thing, Flickr? It was like you could post, if you were an old person and you were doing a safari in Africa, you could post your pictures of zebras on the Flickr. And we had people's ground truth data. We didn't have to go out and look for stripy zebras. We had pictures of zebras taken by professional photographers or tourists, looking at each one of these different habitats, and sure enough, when the temperature changed, you saw stripiness change. The stripes changed. The tsetse fly one is also interesting. The other point that was made about tsetse flies is how many tsetse flies must you have on your zebra body to be like, enough is enough, I'm going stripy. Like, it's a lot of evolutionary change to get to, like, I mean, they also have tails. I don't know if you've seen, they whisk them away. Probably good enough. But we don't know for sure. This is why science is great, because there's always an opportunity to disprove a current theory. But I'm going with temperature. All right. Wait, what time is it? Am I running out? Oh, it's 4:20, great. Let's model several things to environmental conditions. We thought about the zebra one. That's a single thing. This very smart people, Ellis et al., came up with this idea of like, oh, okay, we can model stripiness in zebras, a single thing, or a species across a landscape. Why don't we do this for a whole community of species and see how diversity changes across a landscape? Amazing. So again, no quiz. Also, don't do this. 1, 2, 3. It's very hard for me to follow all this. I can't do it. But here's all those trees that I was talking about. You mash all the trees together for a single species, then mash all the species together, then map all of that, and let's see like what the community turnover looks like. And then Fitzpatrick and Keller, amazing, amazing researchers, they looked at a single poplar species, but they said, hey, instead of species and looking at community composition, let's apply this same method to every individual SNP. So treat every individual SNP like it's a species in your genomic data set. And then look across a landscape and see, hey, how does that genomic signature change across a landscape? So they looked at a single species of poplar. They had 412 reference SNPs and 5 outliers. Outliers being things that looked like they really had an environmental signal, something that matched to environment. And you can actually look at the difference between the outlier map and the reference map. Think about that. Say like, oh, here's a place where SNPs matter and are related to environment versus environments where we don't really see any relationship between environment and the SNPs. And if you look at that map and see the red here, those are areas where selective forces are thought to be playing a very large role. These are places where SNPs, important and linked to environment, are really showing up above and beyond just neutral variation across the whole landscape. So I think that's super cool. If you can do that between outliers and references, you can also, think about it, we developed this really cool relationship between the genome and the environment. If you have environmental predictors and you have a future projection of what those environmental predictors are going to be, you can project this whole system into the future. And so they were able to do that as well, Fitzpatrick and Keller, and say, this is in the future, a future representation concentration, a future climate change scenario. This is the difference between the relationship between genome and environment in the future and what it is currently. And that's shown by this really nice figure here. Look, mean summer temperature is an important variable. We already know this. Temperature can be important. The difference between A and A prime, which is this location currently and in the future, there's a big turnover in the genomic signal as compared to B, this location currently and in the future. Even though the temperature difference is approximately the same, in one location, the temperature change matters, and in the other, it really doesn't, or it's not going to have as big an impact. So all these areas in red here are areas that we really care about under future climate change, because these are places where the genome has to change very quickly in the span of 40 years in order to keep up with the pace of climate change. This is just an amazing sort of breakthrough, and it really kind of changed how we think about like, oh, okay, well, we can predict into the future how this relationship needs to be. We can sort of plan and use that information to inform our conservation decisions. Okay, I'm going to run out of time here, but this is not a mistake. I purposely put this up there because our group was thinking about the same kind of issues and saying, How conceited of us to think that as soon as we figured out a method to project the relationship between genomes and environment, that we think it's starting right now. Like right when I came up with the idea of like, oh, you know what, we should project into the future. Then we start looking and it's like, yeah, now it's gonna start changing. We were like, duh, it's already started. If you look at these these global mean temperature heat bands, you can see the climate had already started changing. So we were like, oh my God, let's look in the recent past and see if we can see signatures of this. And that's what we did. For a yellow warbler, this is the range of the yellow warbler. Again, really not great. Hard to do conservation with this. Breeding range, okay, great. You have a big blob there. How do I do any conservation on that? How do I know which populations are in trouble? Well, it turns out if you look at BBS records for the yellow warbler, not an endangered species, nobody's worried about it in terms of conservation. But look at this map. There is spatially explicit trends such that in the Midwest areas, you have an increase in population trends in yellow warblers. And then along the south and some portions of the northern border of their range, you have dramatic decreases. So we said, okay, let's look at this spatial map of decreases from bird banding stations. Again, no genomics, no anything. We were just looking at population trends. And let's compare that to the gradient forest results. Well, here's the gradient forest results. We found that temperature was very important in this species in driving genomic patterns. And we have this genomic signature across the landscape, or how genomic signal changes with these temperature metrics. And we say, okay, let's do that subtraction and come up with the difference between future and current trends. Here's the BBS trends that I showed you before. Here's the vulnerability map. So when I say genomic vulnerability, I mean that change that has to occur in the genomes of those organisms in order to keep pace with the environmental change. And you don't have to squint to see this. This is a correlation between the places that are going to be vulnerable in the future and the places that we've already seen major declines in these populations. It really spoke to us and said, like, we're already in a time of rapid change for these organisms. And despite this thing being a species of least concern, the yellow warbler being species of least concern, we're already seeing dramatic changes. Real quick, back to the southwestern willow flycatcher, or the willow flycatcher in this southwestern version. You don't have to relegate this to a single species. These are subspecies. The same thing played out. When we looked at vulnerability of the 4 subspecies of willow flycatcher, the southwestern willow flycatcher had the highest genomic vulnerability of any of the subspecies. This population down here has to change more in its genome to keep up with climate change than any of the other subspecies. That's also the population that has suffered massive declines recently in the past 30 or 40 years. We're down to probably about 200, 250 breeding pairs of southwestern willow flycatcher. And the last thing I'll leave you with is the loon, which is not only vulnerable in its southern areas, high genomic vulnerability in its southern areas, but under Audubon projections, just based on climate change alone, we're going to lose that whole southern range. So Audubon, the climate projections under Maxent models are suggesting that we're going to lose that range. Lo and behold, the places that we're going to lose are highly vulnerable, and those individuals, those populations need to keep up with climate change at an unprecedented rate, which is unlikely if their habitat is also being lost. So hopefully I'm kind of imparting to you the seriousness, the concern that we have. I'm working with a number of people on this stuff. If you have data sets like this, reach out to me. If you have questions, reach out to me. We're kind of like in a perilous time, but we need young people thinking about these projects, thinking about how the climate has already changed those morphological characters or those genomes in the recent past, and how are we going to prevent those major changes from causing population or species losses in the future. So I appreciate your attention. Thank you so much for having me. I really feel honored to be presenting to such a group, and come find me afterwards. We'll nerd out about all the details. Thank you guys so much. I don't know if we have time for questions or online stuff.
Probably do a few questions if people want to say.
Oh, 4:30, that was great, perfect. No questions. No, just kidding. Ava had a question? No? What's your favorite bird? Okay.
So with the solar impacts on birds, I don't know that much about it, but I was curious what it looked like a pretty big deal. And so what, if anything, are the solar companies doing? Are they being asked or forced to do anything to mitigate those issues?
Yeah.
I think it's kind of interesting because, you know, I think ostensibly solar companies would be environmentally focused and presumably on board with doing good things for the environment. So I'm interested if they're sort of putting their money where their mouth is.
Yeah, great question. I really went super quick over that, and I apologize. I just wanted to kind of show the application of that. So let's nerd out about solar. The solar industries have been insanely receptive to this. We haven't had any kind of pushback. The solar companies in California, and that was a Department of Energy sponsored project. Department of Energy also gets it. I think we're looking at the impacts of the solar industry under the giant umbrella of like, hey, we also can't burn dinosaurs forever. Like, this is a much better option. But in California, solar companies are required to do a 2-year post-construction monitoring plan. And we're essentially just saying, they hire contractors to go out and morphologically identify things. That's where the 30% misidentification or unidentification comes from. Sometimes these things get singed or scavenged on the property. So it's hard to identify these things. Sometimes they don't have a head, and sometimes, like in the case of a goose, a head is a pretty a key part of identification. So if there's parts of a thing left or a scavenged thing, we can ID it. They've been insanely responsive and receptive. In fact, they're the ones that provided all of their samples. It's on private property, and they've applied for SPUT permits to get those samples to the people like us that can genetically ID them. And we're great. We recommend genetically IDing everything because I need money. No, I'm just kidding. Part of it is that it's a bit of self-interest, but also we're doing better job than morphological identification alone. And it's dirt cheap. I mean, there's a lot of morphological misidentifications. I'm not knocking anybody. Some of these things are really hard to identify in the field. So I've received nothing but amazing support from the solar industry. I would love it if nationwide there was some standard by, you know, a certain percentage of your birds should be, or a certain time of the year you should be collecting birds. And right now those required post-collection requirements are not in every state. So absolutely put the word out that this is a great thing. That paper just came out. You can read it and send me some questions. Any other questions? Yeah, good. Mark. I don't know if you noticed, if you know the people's names in the room, you sound like way professional.
I do? Oh,
No, I do. Like,
oh, you do.
One for
Oh.
Mark.
Yeah. You mentioned in a couple of those instances where like temperature comes out as being really important. Is that, I mean, an actual function of temperature, or is it something to do with temperature affecting the habitat or something within that environment that you could relate that back to in terms of changes in populations?
Yeah, a really good question. We don't know. We get temperature as a good predictor, and temperature might be driving insect abundance, and we don't put insect abundance into the model. So we lose that. And this is when I go back to the statement I said, which is we're making a prediction about what these genomes should look like now and in the future. I'm not making I can't stress this enough. We need good fundamental ecology, field work, and hypotheses. I mean, you will have a job for the rest of your life if you can really critically think about good ecological hypotheses to answer these kinds of questions, because AI is not going to be able to do that. We run a great model. We hope we have the right predictors, but our predictions are always only as good as the predictors that we put into it.
Yeah, and I think people can relate temperature, but I think when it tells them, you know, if you're trying to convince somebody or really show somebody to have, like you say, if it's affecting insect abundance, it's like temperature, what we're doing with temperature because of climate change is affecting these species, and it's like this cascade effect that has, I think, a larger impact on somebody's thoughts or beliefs about
Yeah,
climate
there's
change.
some great examples, and we have done this where we saw temperature as being an important factor. We use that to guide which parts of the genome we looked at, and we saw that those fragments of the genome, you can map those to thermoregulation sites, for instance. So there's a term called gene gazing, which I hate to do, which is people find a Manhattan plot, they get their outliers, and they're like, of course this is related to rodent metabolism. You know, it's like that's kind of exploratory and not really hypothesis driven. So we do try to avoid that, but at the same time, you're hitting the nail on the head. We gotta really think about that, and if you can make that link to the ecology, you know, that's an easy sell to managers and to fish and wildlife. Yeah.
I was just curious if you had shared that data of the pangolin with West African countries.
Oh, yeah. Yeah, we have, we work with ministries over there. It is a long and winding scenic road to have anything change in a lot of parts of the world. So that's a science paper. It made a pretty big splash. Not only there, but in Hong Kong, like trying to figure out, oh, okay, when we do get a seizure, make sure that some of the seizure, a lot of times those seizures are destroyed. Before they're destroyed, make sure we sample. And we work with a colleague that was a co-author on that paper from University of Hong Kong, who's collecting a lot of that data. Most importantly, we try to inform law enforcement in those countries, which again, can be a bit haphazard. But the idea is that if people are aware that these animals are being taken out of the country, I mean, there's places like Gabon in Central Africa that are a testament to how important like ecotourism and conservation could be. Unfortunately, there's countries surrounding that don't necessarily, you know, agree to those same premises. So stay tuned. We're trying to make a best of a bad job. And there's 3 other species there that we really give a shit about. Are we still recording? Give a shit. I can say shit. Who cares? Come on, we're adults.
For the Zoom. Hello. Very engaging, Ryan. Thank you for sharing.
Thank you, Rachel. That's so sweet of you.
I'm not a genetics person, but you are certainly selling this for me.
Okay.
So my question is, what do we need to collect in the field that something can be genetically ID'd or some kind of genomic sequence? And then also, what is the processing time behind those?
Oh, good question. The spatial data is most important, and an accurate spatial data is great. So if you're in the field, I mean, there might be times you're in the field and you don't really think you need to get lat longs of a sample. Just get them. It's so easy. I mean, this used to be buy a $1,000 Garmin GPS and hope it works, and it's cloudy out, and it doesn't. And now you can do all this on your phone. So get a GPS coordinate. Even if you don't think you'll use it now, you might use it later. If you need to get a genetic sample, it varies organism to organism, but it's pretty easy. I mean, a single feather, I've gotten loads of DNA from a hummingbird feather. Like if you get a single feather, as long as it's not cut, if the feather is pulled, that's more than enough DNA to process these things. You can also do a whole genome amplification. We run an identification center at UCLA, and we try to charge, don't record me on this. No, I'm just kidding. It's like between, if you're running high volume, it's like $8 to $15 a sample. If you're running 8 individuals, it's closer to 40 or 50. And that's also paying for like the lab time and the personnel. You know, we pay for health benefits and things like that. So, yeah, but it very much depends on the species. The other thing I completely just blew past or didn't even mention was these methods are getting more and more powerful because more and more people are uploading their sequences. So the way this works is we take a sequence and we try to match it to something on GenBank, which is like this giant repository of sequences. When I started my career like 20 years ago, there was species available, but it was mostly mitochondrial DNA and it was tough to find any matches.